Very good afternoon, and at the outset, I thank Dr. Senthil, Dr. Ravi Keshavan, and then Dr. Kalai Magal, Dr. Shivakumar, Dr. Rajeshwari, and the heads of the other division, faculty and students for giving me this opportunity. So, as mentioned, I work in genomics-assisted breeding for nutritional quality enhancement in maize. But before we enter into our topic, let me take you through that malnutrition is really a big problem because we have around 735 million people hungry worldwide, and around 3.2, 3.1 billion people that do not afford the healthy food globally, and 2 billion people are micronutrient deficient. And if you look into the stunting, wasting, and overweight below 5 years among children, we normally think Africa has more problem than Asia, but it is not the case. It is the other way around. Asia has more problems than Africa. So you look at here, the 52% of the Asian children below 5 are having stunting problem as compared to 43 in Africa. So is the case with wasting. Wasting means the leanness. And then, of course, from overweight point of view, also Asian children are more having more overweight than African children. And this is the global map of malnutrition. We have basically different types of health issues, but of which childhood stunting, anemia in adult women, and overweight in adult women. These are three three most important health issues. And there are countries which has got only one problem, which has got two problems, and also there are countries which has got multiple problems, all the three problems together. The deep orange countries are having three problems together, all the three: childhood stunting, anemia, as well as overweight in women. And our country, India, we have two major issues, mainly the childhood stunting and anemia in adult women. So we have a double burden, we have a triple burden. Burden here means the health related issue. So practically there is no country in the whole world which do not have any kind of malnutrition. Malnutrition is paramount throughout the countries, but the extent and the type of malnutrition is different. Otherwise, every country, whether it's a wealthy or having poor economy, all countries have similar, actually the malnutrition related problems. We all know about the Sustainable Development Goals, which was floated in 2015 by the global community at the United Nations. And there are 15 goals. There are 12. There are 17 goals, of which 12 goals are having the indicators related to nutrition. So if you look at the bar, the orange part is related to nutrition, while the blue part is not related to nutrition. So you will see out of the 7 different goals, 12 goals are having orange bar. So that means nutrition is central to the many of the goals which have been set for at the United Nations under Sustainable Development Goal. And basically the goal 2, that is zero hunger, where we always talk about that we are being agriculturist, we should provide better quality food, higher quantity food, so that nobody is having hunger. Hunger is of two types. One is the real hunger, where we do not have the adequate food. Another is the hidden hunger. Hidden hunger essentially means we have a food, but it is deficient in micronutrients, vitamins, which we cannot see. That's why the term hidden is there. So the zero hunger indicates everything. Like we should produce food as an agricultural scientist, or protect our crop varieties to produce more food, and that food should be also nutritionally enriched. So that is what is very important. So here the main message is nutrition is very, very important to attain the Sustainable Development Goals. Then the very important aspect is our country loses around $12 billion per year due to different types of malnutrition. So, therefore, we should now work together to alleviate this malnutrition. And the silver bullet is that silver line is like if we invest $1, the benefit which we finally get is $16 in any kind of nutrition program. Because if a person is sick, it's not that he or she won't be able to work, so he or she will be a burden to the society, society will not grow. There are many multiple issues. So if $1 is invested on a health problems, then we can get a benefit of $16. So that means we should work towards alleviation of malnutrition. Now, maize is a very important crop. It is an important crop in the world, where around 206 million hectare land is produced. Asia produces 68 million hectares grow maize, and in India we are now having 10.7 million hectare and produces almost 38 million tons of grains. And if you look at the distribution in India, of course, around 60% is used as a poultry or livestock feed. Around 14% is used in different forms of food. 14% used for starch, while around 6% is used as export and other usage. And this is the history of journey of biofortification program in India. We all know that maize AICRP is the oldest among all the AICRPs. So after its establishment in 1971, 3 OPEC-2 based composite Shakti, Ratan and Protina were released. As far as biofortification is concerned, maize is the oldest crop where biofortification program was started, and we came out with the first set of varieties. But of course, we all know that Dr. Wassel and his team at CIMMYT, they combined OPEC-2 with other genes, and they came out with a hard endosperm based variety. They call it as quality protein maize. And several QPM varieties have been released throughout the year, 96, 98, 2001, 4 and all that. But in 2017, we came out with first provitamin A rich maize. Provitamin, although maize is having carotenoids, but the vitamin A level is very low, around 1 to 2 ppm. While the varieties, all the hybrids which we have developed has got 8 to 10 ppm of provitamin A. So similarly, in 2020, 2021 also, we have several provitamin A rich hybrid released from, now it has been released from IRI, then Vipicas. And then in 2022, we had came out with the first low phytic acid based hybrid. We call it as PMH1 LP1. It was developed by IMR Ludhiana. And in 2023, we came out with first triple biofortified maize hybrid, rich in QPM, provitamin A, and vitamin E. So nowadays, the trend is, if you look at the whole path, it shows that we started with QPM, and now we are working with multiple traits. So we have now QPM plus vitamin A, we have then phytic acid, we have then QPM, provitamin A, and phytic acid, so on and so forth. So our present effort is the development of multi-nutrient rich maize hybrids, so that the consumers, whosoever is, should get multiple nutrient at the same time, rather than present in different varieties. All are present in we are trying to bring in one variety, so that by consumption of that variety, we can have those 4 or 5 traits together. Well, as far as the QPM is concerned, we all know that it is the OPEC-2 gene which actually reduces the zyme synthesis, and which is a major protein, which you call it endosperm protein or the zyme proteins. So in QPM, the alpha zymes are more, while non-zymes are rich. Non-zymes are rich in lysine tryptophan, so that is what is the mechanism. And OPEC-2 also affects the lysine ketoreductase, which converts lysine into further metabolites, so that the mutant one, the enzyme produced from the mutant gene, they do not, it doesn't convert lysine into further metabolites, so we have more accumulation. So this is what is the basic mechanism. We all know that. So Vipicase Almora came out with first Mark-IC selection based hybrid in 2008. It happens to be India's first marker resistant selection best product. So they had a hybrid called Vivek QPM9, Vivek Hybrid 9, and now they have come out with QPM version of it. They call it as Vivek Hybrid 9. QPM version of Vivek Hybrid 9, they call it as Vivek QPM9, and it has got 30% more lysine and 40% more tryptophan. Then in 2017, we from IRI developed again 3 marker resistant selection based hybrid. We call it as PUSA HM4 improved, HM8 improved, and HM9 improved. Again, we had normal hybrids which was released. Then we improved it for OPEC2, and all these 3 hybrids have been released through AICRP system. QPM basically means OPEC2 based breeding, but now we are also working with another gene called OPEC16, which is present on chromosome 8. OPEC2 is present on chromosome 7. So what we did is we combined OPEC2 and OPEC16, and it was found that when we combined, we had around 51% more lysine over OPEC2 and 43% more tryptophan over OPEC2. So, so that is what is the greatest advantage. And the another advantage to the breeder is that unlike OPEC2, it does not cause endosperm softness, or we call it as opacness. So the opacness is because of only O2 gene. O16 has no role on the opacness. So that is the greatest boon to the breeders. So now we have number of hybrids which we have on OPEC2 and OPEC16. Earlier the Chinese are working on OPEC16. Now we are the second after China who are working on OPEC16 as well. And this is what the photograph. What happened is we have taken OPEC2 soft kernel. You see there are entirely white endosperm. White means it is soft. The normal one, the orange one, is harder part, but the white part is soft, because even in the normal maize, the inner portion is soft while the outer portion is hard. But if you look at the OPEC16 alone, it is also like harder at the outside while the soft in the middle. So that means OPEC16 alone mutant is as good as the normal maize. We are also working on improving vitamin A, and these are the different colors: white, light yellow, orange, and so on and so forth. And when we profiled our maize germplasm in India, we found around 12%, that is 3% beta carotene and 9% beta cryptoxanthine are having they are present in the entire among the carotenoids. So the major portion, that is 88%, is lutein and zeaxanthin. Lutein, zeaxanthin has no vitamin A activity, while beta carotene and beta cryptoxanthine has got vitamin A activity. We got some symmetline, and we found that 81% of the entire carotenoids are having beta carotene, while the lutein zeaxanthin is reduced to 14%. So the proportion is changed, and this is because of the 2 genes. We call it as CRTRB1 and LCY. 2 classical paper came in 2008 and 2010. One in Science, another is Nature Genetics. So we got some lines from CIMET, and we introgressed that CRT gene. And in 2020, we released a hybrid called PUSA Vivek Hybrid 27 improved. It is an early maize hybrid. It is for the northeastern plain zone. So the original hybrid, that is the blue bar, has 1 ppm of vitamin A, while our hybrid orange bar is having 5.5 ppm of vitamin A after 6 months of storage. So this is what is the first. This is what is the vitamin A rich maize hybrid which we could release. Then again in 2017, in fact, this is the first vitamin A released. We call it as PUSA Vivek QPM9 improved. It is actually first QPM plus Pro-A combination. So it in fact is world's first QPM plus Pro-A combination. It is released for Northern Hill Zone and Peninsular Zone. It is again developed through molecular breeding, and it has got high vitamin A, 8.15 ppm after 6 months of storage, has got high lysine and tryptophan, and of course as a yield potential, it is as good as its original version. So yield we maintained, and we increased the nutritional quality. So it was again developed through molecular breeding, and This Pusa Vivek QPM9, when we harvested in our field, it was covered by Indian Express, and they flashed this story that IRI came out with the world's first ever lysine and tryptophan as well as vitamin A rich maize. We have with us our director, Honorable Director Dr. A.K. Singh, and Dr. B.M. Prasanna. Sir is now heading the Global Maize Program at CIMIT. Earlier, sir was at IRI. The same lab in which we are now working. It was earlier led by Dr. Prasanna. We have Dr. Vignesh, who has done his bachelor's degree from TNAU. And then we have Dr. Rajkumar. So we are the three maize breeders there. Then we also worked with vitamin A and QPM rich hybrid. In 2020, we released again two hybrids called Pusa HQPM7 improved, HQPM5 improved. It has again rich in vitamin A, lysine, and tryptophan. Pusa HQPM5 improved were released for across the country, while Pusa HQPM7 was released for peninsular zone. And if you look at the average yield potential, it is around 6.5 to 7.5 because it is in the medium maturity group. But if you look at the highest yield ever achieved in AICRP trial, it goes to around 10 to 12 tons. In 2021, we came out with another hybrid called Pusa HQPM1 improved, again QPM plus Pro A. So these are, so far, is the market backcross breeding based method. This Pusa biofortified maize hybrid 1 is a pedigree breeding method based maize hybrid. That means it is not a version of any hybrid, but we could combine both QPM trait as well as vitamin A trait, and we could release it for northern hill zone and northeastern plain zone, and of course it is again released for vitamin A, lysine, and tryptophan. In 2022, again we had QPM plus Pro hybrid. These two hybrid, Pusa biofortified maize hybrid 2 and 3, they are independent hybrid. They are not the version of any previous hybrid. It was bred through pedigree method. It was released for three different zones, and it is again rich in vitamin A, lysine, and tryptophan. We also improved the sweet corn. We came out with the two sweet corn hybrids from IRI, and we improved that sweet corn for QPM and Pro A trait. We have 5-fold more vitamin A or around 2-fold more lysine and tryptophan. Now, improving sweet corn for vitamin A is very advantageous because sweet corn immediately after harvest either will be consumed directly within a 2 to 3 days time, or it will be freezed for future use. So under low temperature, under freezing and consumption within 2-3 days, the degradation of vitamin A will be very, very limited. So therefore, if the vitamin A is enriched in sweet corn, we get much more higher quantity of vitamin A than even what we get in case of grain corn. And this is the global map we were part of this review. Dr. Prasanna led this review. This is the global map where the countries with orange color has released the vitamin A rich maize. So in Asia, we have India and China. We have then Brazil in Latin America, and many of the African nations like Mali, Ghana, DRC, and those countries have also come out with vitamin A rich maize. But in India, we released our first vitamin A rich maize in 2017, while China did it in 2019. So we are in fact first in Asia to come out with vitamin A rich maize hybrid, and which was essentially released. We have now a number of vitamin A rich hybrids from India. We are also working on enrichment of vitamin E. So vitamin E is basically tocopherol, and it has got 4 major types: alpha, beta, gamma, and delta. So maize is rich in total tocopherol, but 80% is gamma tocopherol, while 10% is alpha tocopherol. So alpha tocopherol is preferentially absorbed by the receptor in the liver cell, while gamma is not preferred to that extent. So, but the problem is in maize grain, we have only 10% alpha tocopherol, while the gamma is 80%. We are talking about the proportion of alpha and gamma in total tocopherol. So we have a gene called VT4, which converts gamma tocopherol into alpha tocopherol, and thereby we get around 3.2 folds more alpha tocopherol than gamma tocopherol. So we use this mutant VT4 gene, and now we had one hybrid called APTQH5. It has been now identified through AICRP last year, and it has been identified for all four zones except the northern hill zone. and it has got high vitamin E, around 22 ppm as compared to 6 to 8 in normal maize. We have around 6 ppm of vitamin A as compared to 1 or 2 ppm, and we have double the quantity of lysine and tryptophan over traditional maize. So this particular hybrid has got three traits together: QPM, that is protein quality, vitamin A, and vitamin E. So we call it as triple biofortified maize hybrid. We have also improved the sweet corn for vitamin A and vitamin E because we used CRT gene and VT4. So all have been combined. And if you look at the values, the vitamin A has been enhanced from almost like 7 to 8 folds, while alpha tocopherol is enhanced by almost 2-fold. We are also working towards bioavailability of iron and zinc. We all know that maize grains has roughly around 20 to 30 ppm of, or rather 25 to 30 ppm of iron and 20 to 25 ppm of zinc. But the problem is only 5% iron is bioavailable. That means if we consume, only 5% of the iron will be absorbed in our gut, while 20% of the zinc will be only bioavailable. That means 80% zinc and 95% of the iron will go out through our excreta and we will come out of the body. It will not be absorbed. And the real culprit is phytic acid. We all know that. And there are several mutants, LPA1, 2, and 3. So we are working on LPA1 and 2. And now we have hybrids which are in the pipeline, which we combined with the low phytic acid, QPM, and vitamin A. So that, again, our major target is multi-nutrient based hybrid development. So these hybrids are in different stages of trials. We have also, last year, we have also had one hybrid identified through AICRP fodder trial coordinated by IGFRI Jhansi. And we had one hybrid called AFX7. We call it as dual purpose biofortified forage hybrids, where we had good green fodder yield. In fact, this is one of the first 3 fodder maize hybrid released in the system. Along with IRI's hybrid, we had a hybrid from CCS HAU Karnal, or other Hisar, and then GBP UAT Pantnagar. So all 3 hybrids were released together. Through AICRP system, these are the first vitamin A rich, first fodder hybrid. Earlier all have been the OPVs. But our hybrid is the first one, which is also biofortified in the grains. So we have a better crude protein and detergent fiber, quality fiber, higher dry matter digestibility is there. But apart from that, we also have better quality in the grains like lysine, tryptophan, and vitamin A. So this is the first dual purpose biofortified maize hybrid. An obvious question is there any yield penalty? The moment we work on quality and try to stack so many genes for different traits, the automatic question people ask is, what happens to the yield? So these are the hybrids which are the EDVs. The blue bar is the original hybrid, and the orange bar is the improved hybrid. For example, QPM is the blue, and in this it is written as QPM and Pro A. Then we have normal and QPM. That means normal was improved to QPM, and it was tested in AICRP trial during 2014 to 21, the span of 7 years, but for 2 years, in some cases 3 years. And the AICRP data suggest that there is no yield penalty as far as these traits are concerned. Most of the cases, the orange bar is at par with, statistically at par with the blue bar. Although in many cases it is numerically higher, or even in 1 or 2 cases it is numerically lower, but statistically they are at par. So there is no yield penalty. So far in India, we have around 18 hybrids which have been developed through molecular breeding, of which 14 have been developed from IRI, while others in other case, VPKs and IMR have come out with 2 hybrids each. And these numbers is going to increase in number, because many institutes are now coming out with this molecular breeding best method, especially in case of quality enhancement. We have also done some cost analysis, molecular breeding versus biochemical estimation. These traits like vitamin A, lysine, tryptophan, vitamin E, all are to be estimated through HPLC method. And the cost of HPLC per sample is something around 2,000 rupees. So if you really want to develop a hybrid through a backcross breeding method, that means you have a normal hybrid, you want to convert it into a QPM or vitamin A or vitamin E based hybrid. So if you totally rely upon this phenotyping based method, here phenotyping means HPLC based estimation, we would have involved 23 lakh rupees. But if you are relying on molecular breeding, where the segregating generations are screened through markers, we could complete it in just in 1 lakh rupees, because the per sample cost to do a marker analysis is roughly around 40 rupees per sample. We're not talking about the background selection, we're only talking about the foreground selection, but with 2 backcrosses. So there is a great advantage, but of course in this marker selection also, we estimate the HPLC, estimate the samples for through HPLC only at the last stage, when the lines are fixed or ready, not in the segregating generation. So that means this molecular breeding is not only fast, it also saves a lot of money in terms of development of a particular hybrid. So we also gave our vitamin A rich maize hybrid to National Institute of Nutrition at Hyderabad, and they did a in vitro based CACO2 cell assay. We all know that CACO2 cell assay is nothing but it mimics the human gut cell, and it was found that if you consume 200 grams of vitamin A rich maize grains, it can provide 52 to 64% of the RDA. Although 200 grain consumption per day is simply impossible, but it's just an experimental value. But it suggests that that provitamin A is not only present in higher quantity in grains, if we consume, it will be translated inside the body, it will be absorbed, and therefore the retinol level or the vitamin A level in the blood will enhance. We have also done our experiment with the Directorate of Poultry Research at Hyderabad, and we gave our QPM plus Pro rich maize, and they did it with various other types of diet, and they found QPM plus Pro diet requires least feed requirement. Feed is costly. So for a farmer, if the feed is made up of our QPM plus Pro hybrid grains, it requires less feed than compared to the traditional maize, even the QPM maize. So that means combining both the traits has got a great advantage. And they have done this experiment with a Vanaraja bird, and which is a very popular bird throughout India nowadays. This is an international study. This is not our study. It was done by Moreno et al. 2016, and they developed some transgenic maize where they have improved the vitamin A level, and they did that experiment. So, the carotenoid present in the grains are directly accumulated in the skin, the leg shank, beak, and the egg yolk. So, if you look at the left side photograph, the skin color and the leg shank color is yellow, while if it is fed with white maize, it will be whitish. So, if you look at the international market, if you want to go for an export quality one, so the yellow is much better. But here they have studied the egg yolk, the vitamin A level in egg yolk. And if you look at the third one, which is orange in color, it has got 3 ppm of vitamin A in the egg yolk as compared to 0.3 or 0.4 in the traditional yellow or orange maize available in the market. So that means there is 10 times more accumulation of vitamin A in the egg yolk once the chickens are fed with the maize grains rich in vitamin A. So that means it's a great benefit, because what happens once we consume egg, we only think one thing, that egg is the cheapest source of protein. And of course, if the egg yolk color is more orangish or reddish, we think more vitamin A. But actually, it is not the fact. Color has nothing to do with vitamin A. Even orange color, you look at here, the deep reddish type has got only 0.46 ppm, while the orange color has 3.09 ppm. So the egg quality will be enhanced. The farmers will get more benefit with no additional cost, because development of these biofortified maize, their growth, their cultivation is as simple as normal maize. So that means it is a great advantage. Well, a very important message. I just went through this brand's online portal, and the disclaimer is I am not no way advertising this brand, but I'm just trying to show this particular brand is selling eggs, country eggs, farm eggs, organic eggs, the protein eggs at the rate of 12 to 14 rupees per piece. right? And the traditional egg is available 6 to 8 rupees per piece in the market. So that, and these special eggs, which they call it as protein eggs and all, what they do, they add certain say fish meal or certain types of I know ingredients in the feed, so that once that mixed feed is consumed, it the quantity is enhanced. So therefore, if we can sell this vitamin A rich egg yolk, which I have shown in the previous slide, the eggs can be sold in the similarly in the higher price with no additional cost in the feed. Here we have to add something in the feed, which will involve cost, right? But in our case, no, nothing has to be added. Only the simple biofortified maize grains will enhance the vitamin A level in the egg yolk. So there are a lot of scopes, especially in the cities, malls, where people can afford to pay more. So such kind of niche market, these kind of eggs can be promoted. And even in the villages or other places, no need of any additional cost. At the same cost, we can give higher nutrition. We all know the importance of nutrition after COVID. So therefore, a good branding can help in a big way to popularize these biofortified maize. This vitamin A rich maize has also got lesser aflatoxin contamination, which is a major issue in the in India, especially in the coastal area or area having high humidity. Because when farmers stores it in the gunny bag, it absorbs moisture. Aflatoxin is there because of the growth of aspergillus fungi, which spoils the grain. But in this particular case, vitamin A prevents this aflatoxin contamination, and it's an article from CIMMYT, and so therefore it has got an additional advantage. We have developed now several biofortified maize hybrids from ARI, and so far we have signed 26 MOUs with 14 private seed companies and 1 FPO. So now the private companies are also coming up, because to really popularize, it will be very difficult for a public sector organization to produce such a big quantity of seeds. It is simply impossible. Because we do not have that much infrastructure, then branding capacity, and then marketing capacity. So the private companies, we have offered this. I think it is a model with every organization. They can come, they can pay certain amount, and they can sell our seeds in the market of course, they have to keep the name of our hybrid as it is. They cannot change it. So far we have signed 26 MOUs on this biofortified maize. And this Pusa Vivek QPM9 improved, which is a QPM plus Pro maize hybrids, early maturing hybrids. It is a big success in case of in Kupwara district of Jammu and Kashmir. This hybrid gives around 8 tons of maize grain yield, and it produces very high amount of vitamin A, because the people in there are some tribal belt in Kupwara who consumes both maize as a grain, as their part of food, and also they use it in the poultry industry. So we have signed an MOU with Squash Srinagar, and we have transferred our parental line. They are undertaking seed production in farmers' field, and the farmers are very, very happy. And this hybrid matures in 115 days in such a high altitude. And why 115 days? Because of the cooler climate. The other hybrids present in Jammu and Kashmir, they mature in 125, 130 days. So that higher the maturity, it becomes very difficult for drying up, because for them, after the summer season, immediately winter sets in. So they do not have such drying period. So they want this 115 days variety, which is perfectly suitable for their region. And I'm very proud to say that on 28th of September 2021, our Honorable Prime Minister have dedicated 35 special varieties from developed by our NARS system, of which 2 of our varieties, Pusa HQP1 improved and Pusa biofortified maize hybrid 1, which is a double biofortified maize hybrid, was basically dedicated to the nation. And well, we have a lot of limitations in popularizing biofortified maize, which includes, of course, we have developed now high yielding biofortified maize, so good extension capacity, high quality seed production. Then we need to provide possibly at least assurance of minimum support per price, buyback policies, then bringing food processing industry, putting it in different government schemes like in NFSM, ICDS, and midday meal. Such things will help in popularizing this biofortified maize. So with this, I'd like to thank ICR, my director, Dr. A.K. Singh at IRI, Joint Director Research Head. My heartfelt thanks to Director IML Ludhiana, And Dr. Vignesh and Dr. Rajkumar, they are my colleagues, and it's basically entirely a teamwork which helps in providing tremendous support. And whatever we could do, it's all because of the team. And I thank IMR Winter Nursery, various AICIP centers, funding agencies, donor agencies which provided germplasm, collaborators, technical staff, students, and research scholars. And with this, I'd like to thank the entire team for your patient hearing. And my last aspect from this, my presentation is, this is our team. And when pollination starts, we literally, close down our lab. Entire team is in the field, and we have to do pollination. We do pollination. We don't rely on pollination on anybody else, because maize being a cross-pointed crop, it has to be 100% pure, and that too you are working with so many recessive genes like vitamin A, QPM, and all that. So we have to have minimum, full, there should not be any contamination. So a big thank goes to our team for this. And with this, I'd like to end my lecture, and I thank the organizers for giving me this opportunity to speak about our program at IRI. And also, I'd like to very happy to mention here that now, since we have signed MOU with TNAU and IRI, so we are very much keen to work together. And you have a lot of strength. We have also a lot of strength in certain areas. And if we come together, I think we can come out with a much better product. And so with this, I'd like to thank Dr. Senthil, Dr. Ravi Keshavan, Dr. Kalaimagal, Dr. Shivakumar, Dr. Rajeshwari, and all heads, faculty, and students for your patient hearing. So thank you very much. And I'd be very happy to take any questions if at all anything is there. Thank you very much.
Good afternoon.
Good afternoon, yeah.
So actually, I have some questions for two questions I got during the presentation. Like starting, like we know golden rice, where we have taken a gene from maize itself, and the second version of the golden rice to get more vitamin A. So in maize, we are saying that yes, like okay, there is a pathway, but we you have discussed how the provitamin, whatever is there is still less vitamin A which is present in maize content. Can you tell me in pathway wise what might be the reason for that? I mean, any overexpression or knockout or pathway wise, because in rice we say that the provitamin pathway, whatever is there are two steps where there is a problem. That is why we have gone through transgenics. So similarly in maize, what is the issue? And second thing is, you have discussed about a paper from CIMED on aflatoxin, like the provitamin A rich kernels were also less in aflatoxin. So can you tell in genetic level what was the reason? Is there any pleiotropic effect, or what was the thing behind it, genetic basis?
Okay, so coming to your first question, you mentioned about maize has got less vitamin A. So you are talking about the traditional maize, or you are talking about the maize which we improved?
So you told, I mean, before the improvement, what happened?
Okay,
Traditional.
right. So traditional maize has got 1 to 2 ppm of vitamin A, which is definitely less. That's why we did this breeding. In rice, what happens? The first step, which is to produce from GGPP to phytoin. So that step, the gene is called phytoin synthase. In maize genetics, we call it as yellow 1. So that gene is absent in case of not absent in the sense the gene is not active enough to convert that step. That's why they have taken it from maize. In maize, that problem, that particular gene, we call it as phytoin synthase, is functional in yellow maize while it is non-functional in white maize. So our white maize scenario is same as that of your white rice. So that means this phytoin synthase is missing in white maize while it is present in yellow maize. I hope it is clear, the first point. Second phase is, even though in yellow maize carotenoid is there 1 to 2 ppm, it is not because of that gene that we are getting low. So once the beta carotene is produced, it is actually converted into further steps like lutein, zeaxanthin. So we have an enzyme called CRTRB1. It is nothing but hydroxylase, which hydroxylates beta carotene or beta cryptoxanthin in the further parts. So this mutant allele, which is a natural variant, it doesn't convert so efficiently. It converts very slowly. So which is good for us. It may not be good for the pathway, but it is good for the having the higher vitamin A level. Similarly, there is another gene called LCYE, which converts the flux from alpha carotene pathway to beta carotene pathway, right? So these are the two basic mechanisms of enhancement of provitamin A in maize. Now, coming to your second question, that why beta carotene rich maize or vitamin A rich maize is rich in aflatoxin, I mean, it can protect from aflatoxin. This CIMIT study, what they have done, they have taken a normal set of hybrids and they have taken a vitamin A rich maize hybrid. Their vitamin A rich maize hybrid is also CRT gene based one, and they found it has got lesser aflatoxin. The exact molecular mechanism is not known, but they predicted, or they basically discussed that beta carotene, beta cryptoxanthin being antioxidant in nature, so they somehow protect the seeds, and which doesn't allow the fungus growth to that extent. It doesn't make the grains completely free from aflatoxin. It reduces drastically the development of aflatoxin. So that means the growth of the fungi is somehow restricted. But much detailed work is essential as to know how they are working, what is the mechanism. So to call it as a pleiotropic effect, it is still, we are far off from the actual genetic mechanism. But yes, if it is rich in vitamin A, it causes lesser aflatoxin. And I'm sure your maize program is also developing a good number of vitamin A rich maize, which is now combined with QPM. Earlier, a lot of reports were there, articles were there with low phytic acid. Dr. Sentith's lab have worked. And your breeding team is very, very strong. You have come out with an excellent hybrid, Co6, Co8, and so on and so forth. So, I mean, it's just, we have to basically take this molecular breeding. Many times we say that, we need great knowledge and this, nothing. It's just a tool. In our program, molecular breeding is an integral part of the breeding program. There is no separate breeding program. So, it is just a tool to select. If you use this tool, your job will be easy. You have to grow less population, your cost will be less, your manpower will be less as compared to the traditional method, and you can develop the hybrid much faster than the conventional breeding. So yes, we can. I mean, if all centers start working in this direction, I'm sure in a short period of time, a lot of good products.
Sir, I have a few questions for you regarding the provitamin A.
Right.
So, sir, actually, we say that the beta carotene content is photosensitive. So, and it degrades over time. So, when we take it in the scenario of poultry, so we will be harvesting the maize and we will be drying them. So, essentially, it will be in the farmers' fields. It will be exposed to the sunlight and everything. So, how can we prevent that particular degradation of beta carotene when we are going in this path?
Okay, it's a good question. Actually, vitamin A is degraded through heat, even in fact through light also. There will be some oxidative stress and all that. So it is advised that the grain should be dried under shade, not under sunlight, because anyhow under shade also it can be dried very nicely. Number one. Number two, normally after harvest, 6 months is taken as a normal time, so that the grain should reach the farmers or for the poultry farmers as a feed. Now within 6 months, if you consider from the date of harvest, so we found there will be something around 70% degradation after 6 months of storage, right? But at any point of time, the vitamin A level, whether it is after 5 months or 6 months, will be much higher than the traditional maize. For example, traditional maize at harvest has got around 2 ppm of vitamin A, while our vitamin A rich maize has got around 15 ppm or 20 ppm. After 6 months of standard storage, nothing special, the way a farmer deals it, it comes to around 6 to 7 ppm. While the normal maize, which was 2 ppm, will be less than 0.5 ppm. So you will have a great benefit still by 5 to 6 ppm more. That is point number one. Point number two, at IRI, we are working with another gene called CCD1, we found an allele which CCD1 is an enzyme which causes degradation of vitamin A during storage. So don't think if the maize is stored and harvested, no enzymatic activity will be there. It will be there. And there are certain, because we have around 10% or 12% moisture. So under that moisture level also that gene works. And CCD1 gene is the major gene which causes degradation. So we have found an alternative allele which causes lesser degradation. So our breeding method is now working on not only integrating CRTRB1, we are also integrating CCD1 so that it causes lesser degradation during storage. So these are the second stage of hybrids which is coming up in the pipeline. So actually speaking, your question is, I mean, the breeders have started already working in this direction, that tomorrow we'll have a hybrid which has got not only high in vitamin A, will also have lesser degradation during the storage, whether it is because of light or heat. Right.
Yes, sir. Sir, I have one more question, sir. Sir, like I have come across one paper where they said like the beta cryptoxanthin, it is more heat resistant than the beta carotene. So, are there any strategies that we can select the beta cryptoxanthin content instead of the beta carotene content for uh, provitamin A hybrids, which are not that sensitive to heat or light?
Yes, that is a one strategy, but problem is the vitamin A level is beta cryptoxanthin has got half the value of beta carotene, isn't it, in terms of retinol equivalent? So therefore, yes, if it should be enhanced, but if it is at the cost of beta carotene, so because it's a proportion basically, if you increase one proportion, the other will decrease. Yes, it's a good strategy, but one has to select very, very carefully, because higher beta carotene, beta cryptoxanthin not necessarily will increase the vitamin A, because of the fact that beta carotene has got double the vitamin A activity than beta cryptoxanthin. So that part has to be also taken into consideration. How much beta carotene you would like to reduce in place of how much you would like to increase. So that is something is to be looked into. But I don't think anyone is working in this direction. Everyone is working in a total vitamin A activity.